to share some of the stuff that we're doing in the lab in Valencia. As Sergei was saying, I'm a chemist, but in the last, I was started being a catalyst. I was doing organometallics, but I changed to more biofield when I did my postdoc with Ruth, and now I don't know how to define myself. As I believe most of us working in the field of nanomedicine, we are really in the interdisciplinary stage where we need to understand the biology to be properly applying the chemical tools that we have, but always we need to take profit of our chemistry background to be sure to control this chemistry that is so important for the translation. And this is really the regulatory kind of the way we are working in the lab, where we are, let me see that is not going through. We are really taking into account a whole view on a development of therapeutics. We are really motivated on solve unmet clinical needs. We are working mainly on metastatic tumors and also neurodegenerative disorders. And we are thinking that we are being able to help and to increase this attrition rate of in drug discovery scenario by means of using nanomedicine, especially polymer therapeutics, polymer drug conjugates mainly, but we are playing or we are working with systems that are highly complex, so we have to really give an identity of such a complex system. But not only the identity, where you give how many, which is the drug loading or which type of linker we are using, but also we have to take care of the solution conformation, because we are playing with the bio-nano interface, and this is very important because depending on the size, the shape, the zeta potential that our nanosystem show up to the different biological barriers that they need to cross, the behavior biologically will be completely different. So we need to understand which biological barriers we need to cross, and also that keeping in mind that in our final goal is arriving to a patient, and that the patient population is highly heterogeneous. And then we need to understand which of those patients will be the ones that will be benefiting from our nanosystem. So this is a really iterative system where we need to fully control the identity of the compound, conformation solution, taking into account or understanding the biology, characterizing completely the preclinical models to be able to foster the translation to from lab to clinic. So this is really an iterative approach, and we are trying to take it as a whole in the Polymer Therapeutics lab in Valencia. And using this approach, we are really investigating or having research in different areas. Materials as such, but we are not doing materials. We're doing materials. We select polypeptides as our basic nanocarriers, but usually what we do is understanding the pathology and trying to design our nanosystem to really reach across the biological barriers to treat neurodegenerative disorders, like vaccination, immune applications, some topical diseases like spinal cord injury, or also we are playing with some imaging probes. And within all the field of nanomedicine, as I mentioned, we selected really the work with polymer therapeutics, and in particular within polymer therapeutics, that the major difference compared to liposomes, nanocapsules, polymersomes, is that we are really paying a lot of attention to the linking chemistry. So we are always, especially when we are talking about polypeptide drug conjugates with small drugs, that the talk is going to be focused on that. We have key features that we are really taking into account when we are doing a rational design. After understanding what is our molecular target, which barriers we need to cross, we need to really design which is the carrier that we have to use. This carrier should be multifunctional because we need to have high loading capacity because of a small drug. But also we have to select adequately the drugs, and we are really, as many of you know, working always in combination therapy because the molecular complexity of the pathologies is very high, and then not only one drug will be able to stop the disease. So we are playing with high throughput screening techniques to identify which combination therapy will be synergistic. And the most important part in design, and is what differential from other type of nanomedicines, is that we are mainly playing with the linking chemistry. And in a combination therapy is highly important because we can tune the release kinetics of one drug versus the other. And this could really change from having something that is therapeutically valued or something that is not going to work. And finally, depending on the biological barrier that we need to cross, or if it's something that we can really reinforce the passive targeting that we are mainly working with our nanoconjugates, we can add some targeting moieties that in this talk I will not talk about it, but we are using when we want to cross, for example, the blood-brain barrier, we are using AngioPep, because if not, it will not be able for our nanosystems to reach the CNS when after intravenous administration. So in this talk, I'm going to show some examples where we are designing each of these specific features and how it's influencing the therapeutic output. Polypeptides as carriers, that's the first selection we did. And we select polypeptides because we are targeting some of our pathologies, if they are chronic, as they are looking for chronic administration, like Alzheimer's, Parkinson, or neurodegenerative disorders. And therefore, we always look for working with biodegradable materials. They are biocompatible, and as I mentioned, our cargo is a small drug. Usually, we need a high loading capacity because most of the drugs are hydrophobic. This carrier has to be should be biocompatible, safe, and so on. And the polypeptides has already been in the market and in the with many of you know Vicent, Copaxone, or in the clinics, advanced clinical trial. And these examples of polymeric drugs like Vicent is a polylysine dendrimer, or Copaxone is a 4 amino acids polymer. They are really having a different role or a different behavior regarding immunogenicity and microbiome properties compared to the polyglutamate that has been used to conjugate paclitaxel to use systemically. So one of the things that we have to be careful, especially working with polypeptides, as many, and I saw we will for sure listen to the very nice talks, for example, Fresh Lab from polypeptides. We have to really be careful on the administration way and the application we are looking at. The Vivagel is a topical viruside. It cannot be injected intravenously because it's hemolytic. However, DEP technology that is in phase 2 clinical trials, it has exactly the same carrier pegylated. And then therefore we can really use it systemically. So we have Copaxone is the same, it's subcutaneous because it's highly immunogenic. So Copaxone injected intravenously, it will be giving us a septic shock. So the properties of immunogenicity also in the polypeptide is something that we can really take profit of, but we need to understand when we need these materials to be immunogenic. We are working a lot in vaccines, and the carrier can be use itself as an adjuvant, or we can be completely hidden, and we can also use polypeptides to hide the immunogenicity that, for example, viral vectors are given. So we can use them as a stealth properties depending on the amino acid that we are working. These polypeptides are in this table is showing the polypeptides are in clinical trials in advance and also in clinical advances, and there are many differences. And the field is evolving, and more time the classical classification from Polymer Therapeutics, where we are changing from small drugs, proteins, polyplex, and so on, we are really going to more hybrid materials where we are trying to take profit of different type of nanosystems. For example, we have hybrid materials with metal, or we have a mixture with polymeric micelles, encapsulation and conjugation. So we really have different hybrid materials that are really making the weight, and for sure will give really nice promising results. So when we started working with polypeptides, and it's a very old synthesis and ring opening polymerization of N-carboxyl anhydrides, it's been described, but it's a very demanding. If you want to control it and keep in the living character of the polymerization, it's a highly demanding synthesis, because you have to have very clean monomers, you have to have very well controlled initiation process. If not, you can really have a lot of side reactions like gamma secretion or different type of site modifications. So based on a slab method of the chloride rate, we end up with a method of initiator using BF4 non-nucleophilic salts as initiator that allow us to from an NCA, initiate a polymerization that is living, that we can control the starting and the end terminal group. We could do different architectures, and also what's more important for us, it was allowing us to reach high molecular weight materials that with previous methodologies that we were trying, we could not reach for high amounts, like more than from gram to kilo. We couldn't really go for previous approaches that we check, and with this approach we could go for kilo scale and also for high molecular weight materials. This polymerization approach, it was a spin out in a company that now they have demonstrated, apart from having different architectures, and as I was mentioning, they demonstrated that we have high base-to-base reproducibility with different DP, and also it can be produced now in GMP. PTS is really working with major pharma and biotech companies with different polypeptides that are already in the clinic, or in advanced preclinical scenario. So as I was mentioning, and this is important for us, because this is validating that our carriers can really reach, and we are really working on that direction. And we are using PTS really to have a quality control standards for even for the design of our conjugates from the very beginning, to really secure that we could have a benefit at the end and a cost-efficient translation. So we could do, as I mentioned, linear systems, but also we can not only change the size, the shape, and so on with the branch or star-shaped polypeptides. We can also play with the toxicity. We can have different solution conformation. We can trigger some aggregations and self-assembly behavior, and then we can explore different possibilities regarding different half-life in the circulation, or go into different type of organs. So when we start trying to change from a smaller type of conjugates to looking for a greater type of system that they are able to self-assemble to enhance the blood half-life and also targeting different type of cell types, we change to a star-shaped systems, starting from a BTA core that we thought at the beginning that this BTA core was going to drive the self-assembly behavior after looking at Myers papers. But in our case, we use this BTA core as a macro initiator. And the first example that we did, it was 50 glutamic acid unit branches. And in this case, we clearly saw that we could go from smaller to bigger sizes. We were able to self-assemble nicely to upon concentration. So it was a concentration dependent aspect, but also that really we are getting spherical nanoconstruct that we are not having really any order in the central core. So as you see, we tried to understand first what was going on, what type of self-assembly behavior was happening, because it was not a supramolecular assembly from the core. It was really coming from the branches. It was clear for us because apart from, as I say, showing you here that the CMC was around 0.57, with the concentration we can enhance the size, and at the same time we are decreasing significantly the zeta potential, was important for a specific type of targeting. Also, we were able to disrupt these assemblies by the presence of ionic source, like and not with non-ionic like urea, for example. And looking at circular dichroism with a concentration only with a star-shaped system, we had a red ship with this concentration, showing that it was a conformational issue with the branches. And in fact, this is true, and what this is ruled by a phenomenon that is named ordinary to extraordinary behavior transition in the dynamics of the charged-like polar anions, that is based on the contrarion condensation theory in aqueous solutions. This theory that it was reported in, we are lucky really to find this paper from Muthu Kumar. This theory states that the electrostatic potential becomes attractive as the molecules of charges approach within the Debye length. And the condensed contranions are merging, and then in this shear merge cloud, we are forming transient dipoles that are very strong. And this occurs because the adsorbed contour anions and the polyanion, they are really physically cross-linking very strongly because of these typos. The beauty of this system is that playing with the concentration and the ionic strength of the system, and also playing with the hydrophobic hydrophilic ratio and the starting architecture of the unimer, our hypothesis was that we could really tune the final shape of our nanosystems, our nanoconstructs. And in fact, in the thesis of Alex, we systemically tried to study this, and Alex was synthesizing starch polyglutamase with different type of hydrophobicity coming from the core, playing with different levels of phenylalanines, for example, 1, 2, or 3, and also adding some spacers. And we look at a nice correlation of having short branches, longer branches from few amino acids to like 50 amino acids, the example I showed you before, and also having hydrophobic or hydrophilic spacers in the middle with specific peptide sequences. So Alex demonstrated that for us it was really clear that we could move from something that is more like Myers reporting papers before, where we can have a supramolecular structure from making fibers, or up to reaching something like a more globular nanoparticle, as I show you at the beginning with the first example. The flexibility of the branches makes the system collapse and avoids this structure in the core of the system. However, when we have a shorter branch, when we have something that is around 10 amino acids, as is in this case, or 25 as is in the middle, we can have a more supramolecular assembly and more rigid cores, and the hydrophobicity is the one ruling the self-assembled behavior of the supramolecular assembly. And so we are depending on this length of the branches and the hydrophobic-hydrophilic ratio. We really can tune to have discrete nanorods, long fibers, for example, that we are using to establish a project in spinal cord, or the classical globular nanoparticles. So Alex, in his thesis, defined these nice morphology diagrams where many of these things has been already reported, but we believe some of the architectures that were defined from the supramolecular polyelectrolytes were novel, and the having the discrete nanorods coming from the polypeptides will be the first time reported. So this is for us, giving this type of material, this type of behavior, gave us a possibility to have a versatile nanocarrier that depends on the application, the half-life we need, and where to go in the body. We can really go from a fiber, as I mentioned, or to a more globular shape. Not only changing the length of the branches, or the hydrophobic-hydrophilic ratio, also the functionalities that we put in the branches are important. And Fernanda's thesis, she was looking at brain metastasis, and therefore we are looking for visual fiber cross-linking agents to be able to help the mucodiffusion, mucoadhesion properties to go intranasal, for example. But when we explore this, when we try to have the cross-link system and we derivatize our unimer with the thyroglues, we realize that we can go from systems having nanoscale up to nanogels with a microscale, and this is just the same 50 amino acid unit in the side chain, in the branch, sorry, and then different percentage of thyrogroups in the side chain. So we can really tune also not only the size but also the shape in both. And this is important because we can really change the biodissolution and the PK. But before going to a systemic application, as I mentioned at the beginning, these systems are disrupted very easily in the presence of ionic strength, and therefore we need to stabilize them before going to any biological applications. Alex showed that the discrete nanorods could be nicely stabilized by means of hydrophobic drugs, in the case of doxorubicin or camptothecin, because they can nicely intercalate in the hydrophobic core in this supramolecular assembly, and they were really stable under solution behavior, and they have nanorods from 50 to 120 nanometers length. But our preferred strategy, it really relies, and as I was mentioning at the beginning, in a covalent binding. So what we are doing in the lab is we are derivatizing adequately the unimers before the co-assembly. We can put the drugs even here, or just the cross-functional reactivity groups, and we co-assemble, we self-assemble, we entrap, and we stabilize, and we can do this cross-linking using click chemistry that is not biodegradable. You want to maintain the size for longer, or we can use pH responsiveness, redox responsiveness, depending on what is the aim of the final particle. Because keep in mind that we are working with polypeptides, our final construct will be always biodegradable. In this case, examples we will show you will be polyglutamates that are degraded by cathepsin B. So even if the cross-linking agents are non-biodegradable, the final construct at the end will be degraded into endogenous metabolites like as the amino acids. But if we are making cross-linking that are pH responsive, we could nicely have a longer circulation time, because we have around 120 nanometers. When imagine in the tumor microenvironment, that is a pH, acidic pH, we break it, the cross-linking, then we have the unimers that are 4 nanometers, and with this smaller nanoconjugate, we are able to diffuse and penetrate better in the tumor. So we have a dual property. We can functionalize with drugs inside, as I mentioned, so we have a more controlled release kinetics. But if we want to expose the drugs, for example, if we are playing with antigen or vaccination, we want to conjugate them in the surface of the particle or the imaging probe, so we can do this. So this is a summary as I mentioned up to now. So this is type of cross-linked starch polyglutamates. They are able to have quite a versatile range of applications, because we can tune the size, the shape, and also where we put the cargo, starting from the same chemical tools. So we can really conjugate the drug first before the cross-linking. If we are playing with drugs that are not stable and the cross-linking reactions, we have to conjugate afterwards, or we can conjugate drugs that has to be exposed, so targeting moieties or imaging probes, or we can really go to very big materials with playing with nanojets that we are looking at intranasal administration. Once you have things entrapped, as the stabilization of the system is really high. So this is an example in a click chemistry reaction where the cross-link system is not changing the size upon increasing concentration, or in the presence of ionic strength, in the presence of some salts. So this is really showing that this, after cross-linking, you can use them as a drug delivery carrier. And size and shape is very important. It's very important because we are changing how the fate of these nanocarriers. We are changing and introducing changes in the half-life, and for example, you go from less than an hour to 4 days, and also the biodistribution. So all this, in this case, we are polyglutamates. They are anionic, and then they are renally excreted. But the most important part in the biodistribution of this comparison of these three materials, for example, the linear, the unimer, or the cross-link polyglutamates, is that the cross-link polyglutamates with a greater size and greater negative zeta potential was minus 60, we had a strong accumulation in the lymph node. And these materials, after conjugation with antigen in the surface, we are working with Elena Florindo in Lisbon, and we are using them as vaccines for melanoma and pancreatic cancer. But also taking tropism as a feature, and we are also interested in the renal diseases. I'm just showing an example where, as these materials are significantly accumulated in the kidney, so in a very quick project that we did with the Fundación Jiménez Díaz with Adrián Ramos, we conjugated a natural product, curcumin, that is an anti-inflammatory drug, and also is used as a regenerative drug. We use a lot for different things. It's also used for cancer. So it's a quite dirty drug, but you have to really be careful in the concentration. It's really interesting as an anti-inflammatory for acute kidney inflammation diseases. So in this project, taking profit of that huge accumulation of the cross-link system in the kidney, we conjugated curcumin. We have to conjugated curcumin out after the cross-linking because curcumin was not stable in the cross-linking conditions because it really degrades quickly. This is one of the limitations of the curcumin activity as a therapeutic. But after conjugating the curcumin, that we put like a 14 or 15 weight %, and the size, it was around 80 nanometers with a negative zeta potential, we had a nice accumulation in the kidney again. And clearly in a model of acute kidney inflammation where Adrian was inducing this inflammation by injection of folic acid. So they measure the levels in the blood of urea and creatinine, and you see that in the presence of the conjugate, the decrease of the levels was very low. Also working and looking at the kidney itself, you have histology showing that the diminishing of the apoptotic features like a space 3 or tunnel, and also reactive oxygen species like the radical or the oxidative stress pathways, both major pathways in the acute kidney inflammation process, are fully diminished in the presence of the system. And the real mechanism of how it's working, it's a mixture of having accumulation in the kidney, having as a depot, and then a slower release and greater bioavailability of the curcumin. We don't know exactly in which cells we are accumulating, but we are studying now the specific cells where the nanoconjugate is really growing in the kidney, what is very interesting for other applications we are looking at. So, as I showed an example, also as we conjugate curcumin with an ester bond, it was a degradation. It can be released after hydrolysis, but after cross-linking, we can conjugate the drugs with different linking chemistry. We can control the distribution, the type of yielding, and in a non-bioresponsive manner, but also in a responsive manner. We can also do orthogonal type of chemistries. We are doing combinations, and we also can use self-immolative conjugates. I will show you an example. We like a lot redox responsive systems, and also we use protease or pH as the three major linking responsiveness that we are looking at. When we are looking at the activities as an imaging probe, and I'm showing here an example, depending on the design, we can select, in this case, we select non-biodegradable linking chemistry. This was a project, it's a European project where we are collaborating with CEA, with Vicent and Hideaki Nagase in the University of Oxford, and also the company Sanofi. And the coordinator of this project was Boris Stark, he's a protease expertise. So the idea was designing protease-specific imaging probes. So they were partners in the consortium where they were synthesizing peptidic or molecules responsive specifically to specific proteases that were key in specific diseases like cancer or osteoarthritis. And the role we have in this project was to really use the polymer as a homing device. So the idea was using the polymer to reach the pathological target, and once there, keeping the selectivity of the substrate, being able to have the signal. So in our case, we use different linking chemistry, but mostly the one that was before beta, in the case of the methylalkyl-ACE13, the examples I want to show you now, it was a non-biodegradable linking chemistry. So Vicent synthesized a peptide that was a substrate specific for metalloprotease-13, and what we did, it was really optimizing the linking chemistry, looking at the length and the type of the loading and the length of the spacer. The optimized length of the spacer and the loading gave us that the polymer really was even enhancing the selectivity towards the metalloprotease-13 compared with other metalloproteases. And we also changed somehow the biodistribution. As you can here, you have a kidney excretion again. we had a model of osteoarthritis that it was for contortion. It was not a major inflammation. It was a specific model where they were given a lesion in one knee of the animal, and it has the other one as a control. After intravenous administration of the polymer, what we saw is that compared to the free probe, we are able to have a much longer and sustained detection of the metalloprotease 13 in the pathological site, or in the site of the lesion, and that was really able to be selective and detected in a major manner, also quite early. As you see here, with the free probe, we were not able to detect before 6 weeks. With the conjugate, we are already able to detect already at 4 weeks of the lesion. So this was a nice example that the accumulation of the system in the pathological site in a more specific manner after intravenous administration. We believe it was mediated by macrophages in this case, but we need to demonstrate that. It was keeping sustained, and also it was demonstrated by that it was specific to metalloprotease 13. Sanofi had an inhibitor of metalloprotease 13 that is you see here in this graph. In the presence of this inhibitor, we were not having any type of signal at all in vivo. And therefore, this can be used also as monitor on the disease. So in addition, using the inhibitor as a treatment, using the PGA MMP13 probe as a control, we were able to monitor the disease progression. So they was really having optimizing this linking chemistry and the right site of release, we were able to enhance the properties of these protease-specific signal or imaging probes. But as I said, the linking chemistry in this case was not bioresponsive because we need to cleave the substrate, not the polymer. But in the case of combination therapy, we need to really tune when the drugs are releasing. And in the case, we have a very nice example that is really was a blockbuster combiplex. We have the liposome and the combination, showing that it's really important to have a synergistic ratio and deliver to the same site, same cell at the same site, to be able to properly show this synergistic effect of a combination of drugs. In our case, we want to do this, but we are also implementing the linking chemistry to have the release on one drug in front of the other and having this as an advantage. So in one of the projects in the lab in breast cancer, we are aiming to conjugate 2 different drugs, one in the clinical trials acting as a primary tumor, and then we also looking at a drug that they were able to stop mechanism of metastasis. And in our case, we selected the drugs that were able to inhibit the release of tumor-derived exosomes. To identify which of the drugs were synergistic in having from these 2 families, and also in a subtype specific of breast cancer. So we use different breast cancer subtypes and also 2D and 3D models. We implemented a high throughput screening approach because we are part of the EU screen initiative, European initiative, and we identified a panel of combination therapy at a specific ratio for the specific subtype. With the models we are using, we are 2D on 3D. Having 2D or 3D models is very important because 3D models are giving us a very different readout than having 2D. So we have to be very careful because of the trafficking is very different. As you can see here, different conjugates show different IC50 values. And in the lab, we are lucky and we have even organoids derived from patients, and we have more reliable type of clinical ex vivo models to really test our conjugates. So once the drugs are selected, that's also an important feature. The preclinical models also has to be selected as the organoids. We have to move to in vivo. We have 2 models of triple negative breast cancer that are metastatic. We use an immunocompetent 41 model having metastasis already at very early days, and a 231 luciferase model has already metastasis at day 21 and is efficient because it's a human model. So we have fully characterized these preclinical models are the ones we use to understand and to see the effect of the linking chemistry in a combination. So we compare protease versus pH-Labile linkers. So in a combination therapy approach where we have an endocrine and a chemotherapeutic agent targeting postmenopausal women, we compare the protease-Labile linker with the doxo with the protease-Labile linker with the hydrazone. And when we look at the pH-Labile link in the hydrazone, a shorter or longer length linker to look at also fast or slower release of the doxorubicin. After fully characterization of the conjugates, when we have the two linkers as a protease dependence, the first thing we saw is that we optimize the ratio. So the ratio between the two drugs is important. As you can see here, the high loading or low loading is very important. You have a different tumor diminish. And also that having the two drugs in the same polymer is also very important, comparing, for example, this one with this group here that it has a much lower effect. And we have this type of classical distribution. Again, this is not cardiotoxic because we changed the distribution here with a greater accumulation and longer half-life. And we changed to the hydrazone linking chemistry, trying to enhance this 33% of decreases of the model because 41 is a very aggressive model. We really changed to the hydrazone. The hydrazone was more effective than the protease-Labile. It was not cardiotoxic, but the longer linker, what we found is that we show some signs of hepatotoxicity because the release of the doxo was too fast, and therefore we keep ourselves with a shorter hydrazone linker. In this case, because the linker of the doxorubicin was a pH-Labile linker, we were able not only to diminish the primary tumor, but also to diminish the metastasis in the lungs. This is the 41 model, and then you see here a complete decrease in the metastasis. So if we really select the drugs that are synergistic with the adequate linking chemistry, we can really tune and enhance not only primary tumor in this case, but also metastatic progression. And just to finalize in the last 3 minutes, an example that the linking chemistry is also very important in another type of combination therapy where we are playing with combination therapy using stem cell cultures. This is a project with Victoria Moreno, is a collaborator here at my institute in a spinal cord injury. She is expert in using ependymal stem cells, that is, they are cells that are taken from the same spinal cord, and they are able they are part of this, they are secreted in the cellular spinal fluid, and also they are able to differentiate spontaneously, or you can really drive them and differentiate in different type of active neurons. A spinal cord injury is a very complicated disease where we need to have multiple type of technologies to be able to solve it. And we are
2 minutes more.
Sure. We are able to have to combine not only nanomedicine, but also a type of therapeutics. So we have preliminary data with the curcumin, but we realized that ROCK inhibitors were important for inducing neuroregeneration in spinal cord. And we use fasudil, and again here we designed different linking chemistry, and only a self-immolative linker by redox reductive. It was because it has a major release of the fasudil. We were able to have nice results with the neurite prolongation, and also in organotypic cultures. In an in vivo model of spinal cord contortions, they were able to really reproduce the fibers, and in vivo we were able to recover in combination with the cells in a chronic model after 2 months. The pre-incubation of the conjugate with the stem cells, they allow the cells to engraft beta and to cover beta the lesion and to recover the fibrils. So this is really after intrathecal administration of the cells. And now what we are doing is playing with the fibers instead of the globular system, also to have the support, the mesh that the spinal cord is needed. So that's the summary take-home message. So the rational design of all the components of a polypeptide-based conjugate is very critical for the therapeutic output, and an iterative and multidisciplinary approach is always needed. So thank you very much for the people who is doing the work, not me. I have a really nice bunch of people, from chemists to biologists. We are talking a lot with clinicians. I have very nice collaborators and the funding agencies. Thank you very much also for your attention, and I'm happy